Then guess what? Or 2 less side cancers. Then one's going to be 3 less side cancers.
Learned a little bit.
Are you guys sitting here? Like over here. I'm not going to the volleyball game. I'm coming, so just write my name down.
Never said honestly. On Monday, your heroes can go to a stacked event. Like your team is like
Even if it's no, I'm going to.
I don't.
No, we're going.
I'm not going.
We're not going home.
Oh, that was free. She's not here.
I am so. Yeah, I want to get rid of that.
I'm so mad about it.
I am mad. I don't want to watch anything. We already looked 800 things.
All right, guys. Good morning. Good morning.
Morning.
Time to wake up. All right. Okay, take a quick attendance. Riley. Here. Brianna. Here. Drew. Maria. Anna. Nick. Abby. Here. Hannah. Here. Rosa. Here. Matt. Here. Jillian. Mia. Annie. Brianna H. Brandon.
Here.
Sadie. Here. Jessica. Here. Every time I say Sadie's name, Siri responds to me. Jackie.
Here.
William. Marissa. Anna. Sophia. Aubrey. Matthias. Colin. Olivia. Here. Brayden.
Here.
Amber. Here. Kennedy. Ben. Kendall. Here. Emily. Here. Lauren. Here. Jen. Here. Isabel. Here. And Corwin. Okay. Very good. So today we're going to be settle down, pay attention. You guys ready? Okay. So today we're going to be finishing up chapter 10. So a couple reminders. Your next exam is in a week, next Friday. It's only going to be on chapters 6, 8, and 10, so it's less content, because we did get to this exam really quickly. But that way you won't have anything to study for over spring break, and then we'll just start with the last 2 units after spring break. Your worksheet 4 is already posted. It's going to be just on chapter 10. It's 10 questions. It's more in depth. So it's going to be due Monday. You'll have the weekend to work on it. We'll go through it on Monday. So ask your questions on Monday, and then on Wednesday will be an exam review day. So next week there's no more new material. This is the last day of material for Unit 2. And then Friday is your exam. Okay? So we ended on, I think it was 23. Yeah, so we ended here. We talked about how having glucose present will prevent lactose from getting in the cell, and What's the enzyme that gets lactose into the cell? Yeah. LAC-Y. Does anyone remember its name? Literally on the board. Permease. Yeah. Because it allows lactose to permeate in the cell. So if there's glucose around, then the LAC-Y permease cannot work, it's inhibited, and so it doesn't allow lactose to get into the cell. So the LAC operon is used as this general model for operon control, but there are many, many other ways for bacteria to regulate. So there are some proteins that actually have dual regulatory functions, which means that those can act as both an activator and as a repressor. And another word that we'll come across is attenuation, which means that you can essentially regulate gene expression by coupling transcription and translation. So we'll talk about an example of attenuation. Basically, the ribosomes that are working on translation, they can send chemical signals, and there's chemical signals in the environment that affect whether a ribosome can continue gene expression and translation or not. So we'll talk about that, an example of that shortly. But first I want to talk about the arabinose operon. So far, we've talked about the LAC operon, and now, so LAC operon basically encodes genes that break down lactose. Now we're talking about the arabinose operon. Basically, it has genes that will convert arabinose into an intermediate in a biosynthetic pathway. So it's important to, remember that LAC operon is a catabolic. It encodes catabolic enzymes that will break down lactose. Because we'll talk about anabolic operons, those that encode enzymes for the buildup of molecules, amino acids. But the arabinose operon is somewhere in between, where essentially it converts arabinose into an intermediate that's important. And what you need to know is that arabinose is an important carbon source. But the arabinose operon has a regulatory protein that has dual activities. It can be an activator and a repressor, and that is called ARA-C. Okay? So ARA-C can repress or activate gene expression, and it does this depending on whether arabinose is available. So when arabinose is absent, you have this elongated version of the ARA-C dimer. So it's a dimer because it has two components. And so when it's in this elongated form, remember, everything has to do with conformational changes. Structure determines function. So when arabinose is absent, then the cell is not going to want to express those genes that are going to convert it into that intermediate. So this dimer is elongated, and it represses the expression of those genes that break down arabinose. When arabinose is present, you see in blue, it can bind, and then the ARA-C dimer undergoes a conformational change, becomes compact, and then it can stimulate the RNA polymerase binding to the promoter, which then can transcribe the genes. So it's just a change in conformation that makes the ARA-C regulator a repressor versus an activator. And that conformational change is dependent on whether arabinose is available. Does that make sense? Okay. So just another quick description. Basically here, without arabinose, this is just an explanation of, basically the bonding and just the interactions of why it's elongated, and then it looks like this when arabinose is there, and then you have this dimer kind of looped together. So the arabinose operon is also called the ARA-BAD operon. So basically this is how it's regulated. So this is the operon. And so this is the promoter area where era bad would be translated. So here there's era B, there's going to be another one that's A, there's another one that's D, that's not shown here. So the era C dimer, when there's no arabinose, it basically is in this form, and it binds on this area just to the era. You don't have to remember era I1, era I2, and era O2, but basically in this conformation, it basically keeps the transcript apart, and it doesn't allow the RNA polymerase to bind. Whereas when arabinose is present, then it can change conformation, and it can bind to both of these, and then this will allow the RNA polymerase to bind to the promoter and induce transcription of those genes. So it's all about conformational change. So normally you might be seeing a common theme here, that when things are forming a loop on a transcript, that is going to hinder transcription. So once again, you guys should know this, the era C regulator is this dual function regulator, and when arabinose is in the cell, there's no need for those enzymes to catabolize it. When arabinose is present, then you're going to transcribe the catabolic enzymes that will break down arabinose into that intermediate. Okay. So, like I said, the lac operon and the arabinose operon are both operons that encode enzymes for the catabolism or the breakdown of lactose or arabinose. So, we'll also talk about anabolic or biosynthetic pathways. And so the repression of these is pretty different from repressing a catabolic system. So the reason is, so say for instance here, this is the tryptophan operon. Tryptophan is an amino acid, and we want to make amino acids. Our cells want to make those amino acids. So this is an anabolic operon where you have different tryptophan genes that encode enzymes that will eventually make tryptophan. So if you're making tryptophan, you start with very little and then you have a lot in the end. So it's different from catabolism. Eventually, you have enough tryptophan in the cell, and it has to eventually turn the anabolic operon off. So typically, the way that you turn off these operons that are going to make the amino acids you need, they're usually regulated by what we call inactive aporepressors. So these genes, there's a new term that you guys will have to know, the aporepressors. Basically, the way this works, and I'll show you the actual picture demonstrating this, but this is a good summary. You have an aporepressor that will bind a co-repressor. The co-repressor is the end product of the biosynthetic pathway. So can anybody guess in the tryptophan operon, for making tryptophan, what would be the end product of the pathway that would tell us we're done making tryptophan? Yeah.
Presence of tryptophan.
Exactly, yeah. Tryptophan would be the co-repressor because that's the end product of the pathway. So if we've made it and there's a lot in the cell, then that's going to activate the aporepressor. It's inactive right now, but then when there's enough tryptophan, which is generally the co-repressor, that's going to be able to bind the apo-repressor, and then that will halt transcription. So this is called a negative feedback mechanism, because the end product of a pathway now is used as a co-repressor to repress the expression of that operon. Great. Any questions so far? Okay, so how does this work? So let's just get ourselves familiarized. So you have the trip operon, you have an apo-repressor, this is usually inactive, and then the co-repressor in green, that's the tryptophan, basically we write it as just trip. So if this operon is able to transcribe these genes, makes these proteins that will make tryptophan. And so when tryptophan levels exceed the cell's cellular needs, when it's present, then it can bind to the apo-repressor, which in this case is TRPR, trip repressor, and then the co-repressor is just trip. And so once they are bound, then this complex can bind to the operator sequence, and that will then block the RNA polymerase and repress expression of those genes. Okay, does that make sense so far? Pretty straightforward, negative feedback loop. But if you'll notice that there's this in orange right here, just this little region called the attenuator region. And so I've talked about this a little bit, but basically repression is not the only mode of regulation for this TRP operon. A lot of amino acid biosynthesis operons use something called transcriptional attenuation. Basically, they use the ribosome as a sensor of the amino acid levels in the cell. So if you remember, for a ribosome to work, what does it need? What needs to come into the ribosome for a ribosome to be able to translate a protein? It's a type of RNA. Yeah.
Amino acyl tRNA.
Exactly. You need the amino acyl tRNA, or the charged tRNA. That tRNA is charged with an amino acid. So for a ribosome to work, it needs the charged tRNA, and the tRNA won't be charged efficiently if you have low levels of that particular amino acid that you want to make. So eventually, you'll get ribosomes that will get tRNAs coming in that are not charged. So now the ribosome can be used as a sensor for what amino acids the cell is depleted for, and this plays a role in this concept called attenuation. So you can also think of it as a transcriptional control mechanism. So over here, we have the attenuator region. And so if you kind of zoom into what it looks like, there's these two loops. You don't need to know all the details here, the 1 to 2, 2 to 3, 3 to 4 loops. But what you should know is that there are essentially two codons that are in the tryptophan operon that are able to form these stem loop structures. And basically, the ability to translate part of the mRNA determines whether that transcribing RNA polymerase will continue to make that transcript. So here we have a leader peptide. And so basically this will affect whether things that are downstream are able to be transcribed. So it's a mechanism in which the ribosome is able to sense what amino acids are in the cell based on the tRNA being charged or not with the amino acid. So just looking at the mechanism here, if you have high tryptophan levels, the ribosome can translate through those codons, and then eventually it will encounter a stop codon that tells it you can stop the translation. So the ribosome stops over here. It's at this region. And so then it can continue to translate this part. And so so now the RNA polymerase is here, and so it can continue to translate, and then it eventually will realize that, hold on, do I have this in the wrong order? Okay, so yeah, I had that right. So basically, you have the ribosome here, and it's translating, and then at the same time, this RNA polymerase is trying to transcribe the DNA into RNA. So when you have low levels of tryptophan, then you know that you need to make more tryptophan, so the ribosome here will stall. It's not able to continue doing translation because it realizes that it needs tryptophan, and so because the ribosome stalled, it's basically telling the RNA polymerase to go on and continue to make the transcription of the genes that are supposed to make tryptophan in the cell. So when you have high tryptophan levels, then it halts transcription of those genes because the ribosome says, well, there's enough tryptophan, the charged tRNAs are coming in with the tryptophan, so we don't need to continue making RNA, we don't need to continue making the mRNA by the RNA polymerase. Does that make sense? Did I confuse you guys more? A little bit. Okay. So the reason for this essentially is attenuation will halt transcription that's occurring in progress. So it helps to provide a quick response to changing amino acid levels in the cell. So it's not like repression because the ribosome now is involved in sensing the levels of the amino acid. All right. Any questions here before we go on to the next section? All right, so we talked a little bit about quorum sensing. It's a really, really cool phenomenon where bacteria are able to communicate with each other, and they do this by having these conversations based on chemicals in the environment. So this is another type of gene regulation, but here bacteria are able to signal not just things that are occurring intracellularly, but it also allows for the population of bacteria to communicate with one another extracellularly and work together. So basically, there's this cool discovery that I've mentioned previously in Vibrio fischeri. It's a bioluminescent bacteria that can colonize the organ of a Hawaiian squid. And it basically allows the squid to either hide, camouflage, or not, depending on the environment it's in. So when you grow Vibrio fischeri on a plate, it looks kind of similar to what E. coli would look like, but then if you turn the lights off, it glows. It's really cool. But it doesn't express this molecule that allows it to glow, the bioluminescence, all the time. Basically, it doesn't make sense for one single cell to turn its flashlight on It only does this when it can sense that there's a lot of the bacteria around it that also can do this, and then they all turn their flashlights on when there's enough cell density in that population. So if one bacteria turns the flashlight on, you won't see anything, essentially. But instead, these cells are able to release these molecules called autoinducers, and they can actually measure the concentration of that autoinducer in order to determine that they're at a high enough cell density to turn these sets of genes on. In this case, these are genes for bioluminescence. So, this triangle here is the autoinducer molecule. If there's, one cell, it's low cell density, there is this molecule being made in the environment, but then when you have a high cell density and they're all making this molecule, then this signals to the cell that we need to now transcribe genes for bioluminescence so that we can all turn our flashlights on together. And so this is a graph where you're looking at over time. These are arbitrary units because they're looking at two variables, biomass, which correlates to cell density, and then bioluminescence. And so you can see that, this bioluminescence is pretty much at like very steady, very low levels. Nothing is really turned on here. And then as soon as you reach a certain biomass, a certain cell density, then these they know to turn on these genes, and they can then turn their bioluminescent on. Does that make sense? So remember, this is in the extracellular environment. So how does this work when you're thinking about it inside the cell? Inside the cell is where the genes are that need to be turned on for bioluminescence. So basically, the autoinducer is outside of the cell. And so once there's a certain extracellular concentration that's met, then it has the ability to re-enter the cell, and then the autoinducer accumulates, can diffuse back into the cell, and then once it's high enough, then it can bind to LuxR, which will relieve repression of the operon by kicking off the repressor. So LuxR is a repressor in this. So generally, at low levels, there's something repressing these bioluminescence genes. So there's a repressor that's bound. So once there are high levels of the autoinducer in the environment, then that can diffuse back into the cell, and then that will bind, the autoinducer binds to LuxR. It kicks it off, and then that relieves repression, and then you can make the luciferase enzymes, which then can make that light property of bioluminescence. Any questions? So let's see if this video will work. I had no sound earlier when I tried it.
Off the coast of Hawaii lives a cunning nocturnal hunter with a brilliant trick up its sleeve, or rather its mantle. Meet the Hawaiian bobtail squid. After spending the day sleeping buried in the sand, come nightfall, the little squid emerges in search of a meal. To hide itself from predators and prey alike, the squid has its very own invisibility cloak, a colony of a glowing bacterium called Vibrio fischeri, which it houses in a special organ in its mantle. In an ingenious form of camouflage known as counter-illumination, the squid uses its glowing bacteria to mimic moonlight, hiding its shadow from any creatures looking for an easy meal. Moving through the depths, the squid uses its ink sac to adjust the intensity of its living torch to match the strength of the moonlight. Also handy if a cloud suddenly passes in front of the moon. Vibrio fisheye glow because they have genes for producing light using a simple chemical reaction. Scientists growing Vibrio fisheye in the lab notice something quite interesting. The bacteria only switch on their light when they have reached a critical density. This makes perfect sense. There's no point going to all the trouble of making light if it isn't bright enough to be seen. But how do they do it? Each bacterium produces a chemical signal called the autoinducer. The more bacteria there are, the more autoinducer there will be. If those bacteria are growing in a confined space, like a flask, or the light organ of a squid, the amount of autoinducer will start to build up. Once the autoinducer reaches a critical concentration, it triggers the bacteria to switch on the genes for producing light. This chemical form of communication is called quorum sensing. It turns out that many species of bacteria use quorum sensing to coordinate their behavior, from sex to swimming to causing disease in plants, animals, and humans. Scientists hope that one day quorum sensing may be exploited to turn the tables on some of our deadliest foes. And if they do, it may well be thanks to that little nocturnal hunter, the Hawaiian bobtail squid.
Okay, does that help give a little bit more context? Pretty cool. So, one of the pioneers of this field is Dr. Bonnie Bassler. I did have this on your last study guide, just her TED Talk. I don't know if you guys saw it, but it's really, really cool the way she describes it. But bacteria basically use quorum sensing for both universal communication as well as these secret conversations in the same bacterial species. So, we have these intra-species communications with the same bacterial species, so if it's all Vibrio fisheye, they can count essentially their siblings. Autoinducers kind of gives this specificity for species, there's different bacteria that can produce different autoinducers. So they're related, but they're still different. So the AIs, or the autoinducers, are sort of this language of these different bacteria. So, why do bacteria do this? It's not just for light. Many pathogens and bacteria use quorum sensing to control virulence genes. So virulence proteins, does anybody know what a virulent gene is? Basically, a virulent gene is a gene that allows for some pathogenic potential. So it could be a toxin that's produced by a cell, it could be a protease or something that's going to degrade things. Usually a bacteria can control the expression of these virulence gene to control them, across the entire population using quorum sensing. So once again, they're not produced until the density of the cells is really high, and it can overwhelm its host. And so if a pathogen is trying to, infect a host and try to, cause an infection in the host, then it can turn on these virulence genes, but only when it knows that there's enough bacteria of the same species that are there for them to all turn on, these virulence genes at the same time. So they use it for protection, but it's important to remember that bacteria live in diverse communities. So do different bacteria also talk to each other? So far we're talking about intra-species communication, but quorum sensing is also used to communicate with other species. So this is called interspecies communication. There are universal autoinducers that several different bacterial species can recognize and respond to. So basically, we have interspecies and intraspecies, and it helps them know, like, do we want to put up a fight and all kind of get together and do our thing, or do we want to just lay low? So it helps tell the bacteria whether they're the majority or the minority. And this has actually been a new target for antibiotics, where we've used antibiotics to trick the bacteria into thinking that they're always in the minority, so they never express those virulence genes. Any questions so far? So if you guys have heard of like a virulent strain of something, it means it's a strain that can cause disease. Okay. So far we focused largely on these individual gene circuits or operons, but there are several behaviors of organisms that require these integrated gene circuits. Basically, it's just more complex decision making. One of those is chemotaxis. Basically, a bacteria has to know when it needs to swim and in what direction it's going to swim towards. Is it going to be swimming towards a chemoattractant, a chemical that's an attractant for it, or is it going to swim away from a repellent, like an antibiotic? So many bacterial genes are regulated by a hierarchy of different regulators that form these integrated circuits. So, these gene switches can regulate these decision-making processes really quickly. So should I move? If so, where? This is called chemotaxis. So we talked about bacterial flagella a little bit, and that is essentially that like tail at the end of some bacteria that allows it to move. And we talked about, that the flagella have this flagellar motor that has to spin, and then that allows it to move. But for those that use flagella to move, they don't simply dash about hoping that they swim into a favorable condition. There's a regulated circuit, an integrated gene circuit that's very regulated, and that's chemotaxis. It's the ability of an organism to sense chemical gradients and modify its motility and response. Motility means movement. So they can either swim towards a favorable environment, which we call a chemoattractant because it's a chemical signal, or away from an unfavorable environment, so a chemorepellent. And this use of the flagellar motor, it's not random. It does have direction. It can go counterclockwise, which is more of this directed and smooth swimming, and then clockwise results in tumbling. So basically it's this biased random walk in which sometimes, the cell can move a little bit randomly, but overall they tend to migrate towards the attractant or away from the repellent. So if you look at A, we have this counterclockwise rotation, and so when it's doing, the counterclockwise rotation, it's more smooth in motion and it can move towards the attractant in this case. Counterclockwise movement rotation, now these are kind of all going in their own counterclockwise direction, and so that causes it kind of to tumble. So if there's an attractant right here, you have this bacteria, moving in the counterclockwise, it's going to swim towards the attractant, but then once it gets close to the attractant, it might, kind of just start to randomly, like there is some random movement in there, so then it might kind of bounce back a little bit, and then it realizes, okay, I need to move back this way, so it starts moving clockwise, and then it tumbles back in this direction, and then once it gets back in this direction, it realizes the attractant is still right there, so then it swims toward it using counterclockwise. So what you guys need to know is, the directions, the rotations, and what type of movement this does. You should know what chemotaxis is and the flagella. And so a lot of bacteria you'll see only have one flagella at the very end. This one has multiple, so there are some that have flagella all over, and that, you know, definitely influences some of the orientation, but this is the general thing for counterclockwise versus clockwise movement. If you notice, there are receptors for the attractant, so eventually those receptors will bind the attractant. So this is an FYI slide. I don't need you guys to remember this, but it kind of does depict what exactly happens. So you're welcome to read this on your own. We can go through it on the side if you want, but just an FYI slide, because the E. coli does this, and we work with E. coli in the lab, so sometimes it's cool to kind of know a little bit more about the mechanisms behind things. Are there any questions? Okay. Have a good weekend. Worksheet 4 is due on Monday. Bring that to class. If you won't be here, then send it to me via email on Monday, and we'll do a review.